energy from a high energy compound is used to make the ATP. And that is called substrate level phosphorylation.
He does not care about it. That's almost perfect. More than perfect. 100%. Then you got a 40
ATP, of course, will be made by phosphorylation. You are adding a phosphate to ADP. And so that's why it's called phosphorylation. And there are two ways of doing that. One is by substrate level phosphorylation, which basically means that you have a high energy compound, and in the reaction that energy is transferred to a ADP phosphate bond. And the other way that ATP is made is by oxidative phosphorylation. That means a reducing agent will be oxidized by oxygen. And whenever anything is oxidized, it releases energy, and that energy is used to make ATP. We also saw several oxidation steps in glycolysis. Not several, just one. But we are going to see many more in the citric acid cycle. And that also gives you energy, and that in the very next step, that was used to make ATP. And also it made 1-NADH. So when something is oxidized, it actually is equivalent to a lot of energy, and usually ATP can be made, and at the same time you also produce a reduced species, which was the NADH. So always remember that NADH, when it is oxidized later, that we will do after the citric acid cycle, it gives you 0.5 ATP per NADH. So that's a lot of ATP compared to, for example, substrate level phosphorylation, which gives you only one. The whole of glycolysis gave you two. And there will be numerous NADH that are formed in the various reactions, and each of them will later give you much more energy. So most of the ATP comes by oxidative phosphorylation. For example, from one glucose you are supposed to get 32 ATPs, and only 2 of them you get from glycolysis, and the remaining you will be getting from oxidative phosphorylation. Not all the remaining, that we will also see in citric acid cycle you again get 2 from substrate level phosphorylation, but the remaining 28 will be by oxidative phosphorylation. So always remember these two ways of getting ATP, one by substrate level phosphorylation. Whenever you have a high energy compound, for example, we saw phosphoenol pyruvate gives you ATP, and then we also studied that thioesters are high energy compounds. They will also give you ATP in the citric acid cycle. So in the citric acid cycle, as I said before, the cycle of course starts with acetyl-CoA, which is a 2-carbon compound. The acetyl group is 2 carbon, pyruvate is 3 carbon. So in order to go from 3 carbon to 2 carbon, there is another step. And that step is actually a very difficult step to do, but no one bothered to name that as any pathway, because as I said, pathways are named by people, it doesn't really mean anything. So this doesn't have a name, it's just a reaction that is between glycolysis and the citric acid cycle. And because it happens in the mitochondria, it is oxidative reaction, and so it is included along with citric acid cycle. And one more time, I explained before that aerobic cycle doesn't mean, or aerobic reaction doesn't mean there will be oxygen as a reactant. It means that it is dependent on oxygen in future reactions. Unless those reactions happen, then this one cannot also happen. So the whole of citric acid cycle, we will find there is no oxygen anywhere. And including this reaction, pyruvate to acetyl group, there is also no oxygen involved. But then they are still called aerobic reactions because later on the NADH must be oxidized to NAD to continue these reactions. We saw the same thing happen in glycolysis. That was a question in one of the quizzes, why is pyruvate converted to lactate? And the reason is that you run out of the NAD and you need to get the NAD back, and that's why pyruvate is converted to lactate. in the mitochondria, the NAD that is used up also needs to be converted back to NAD, otherwise the cycle cannot continue. And in that case, the NAD, there is no other reaction like the pyruvate converting to lactate because there is nothing to react with. Both the carbons are removed as carbon dioxide, there is nothing left. All we have accomplished in the citric acid cycle is to make NADH, and only way your citric acid cycle can keep going is if you oxidize the NADH. So after citric acid cycle, you need to oxidize the NADH, otherwise more citric acid cycle cannot happen. More of the pyruvate cannot be oxidized to acetyl group, and that's why these are aerobic pathways, even though there is no oxygen involved. So, pyruvate is a alpha keto acid. As I mentioned before, beta keto acids are very easily decarboxylated. So in order to go from C3 to C2, you need to remove a carbon dioxide, and there is a very easy carbon dioxide you can see here in the carboxyl group, and that's why this is a decarboxylation reaction. But decarboxylation of alpha keto acids is very difficult. Decarboxylation of beta keto acid is very easy. And that's why enzymes that do beta keto acid decarboxylations are smaller enzymes with faster reaction, and those that do decarboxylation of alpha keto acids are huge, consisting of multiple enzymes, multiple coenzymes, and a lot of things to study, which we are not going to study that much in detail, but the whole thing is very complex. So one more time, if it is a beta keto acid, let's draw that separately. So this will be a beta keto acid, and the reason this is easily decarboxylated, in fact you don't even need an enzyme, these are decarboxylated by itself with slight increase in temperature. So this forms a bond here, and this bond breaks, and these electrons go to the oxygen. So you can easily draw these curved arrows showing that the electron of this carboxylate ion eventually is going to this keto group, which is at the beta position. And that's why this is called an electron sink. So this electron goes here, and that's why it is decarboxylated. That would happen, of course, with alpha keto acid because you don't have this extra carbon in the middle. And so alpha keto acids are decarboxylated with difficulty, and so it needs a coenzyme. So let me write the overall reaction first. So this gives you, so this carbon dioxide is gone, and then you have and this is the acetyl group, CH3CO, and this will be the coenzyme A, which, and the coenzyme A has a SH group, as I mentioned before. And so it is also written as CoASH, so you may want to write an S here, but it is understood that the bond is always to an S. Alright, so this is the overall reaction, but in this process, many things are happening. This carbon dioxide is removed, and there are coenzymes. There is a coenzyme called thiamine pyrophosphate. This thiamine is not the same as thymine, Thymine. If people have taken molecular biology courses, you know DNA has 4 bases. One of them is thymine, that's different. That's a different spelling, THY. This is THIA, and this is a vitamin. This is actually vitamin B1. It's derived from vitamin B1.
Seductively pulling my iPad out of my bag.
And then there is another vitamin that is used as a coenzyme here, and that's called lipoamide. There is another vitamin, it's a vitamin and a coenzyme, which is FAD. And then finally it is NAD. As I mentioned before, anytime you have FAD, there will always be NAD somewhere involved because FAD is a prosthetic group, and when prosthetic group is the oxidizing agent, it will become FADH2, and that will not leave the enzyme. If it doesn't leave the enzyme, then it cannot be oxidized by oxygen. Because remember, your eventual energy has to come from reaction with oxygen. FADH2 cannot be getting out of the enzyme, so there is always another enzyme that will transfer the hydrogen from FADH2 to NADH, and NADH is a co-substrate. So once again, understand the difference between prosthetic group, which is always attached to the enzyme, never leaves, and a co-substrate which leaves the enzyme, goes to another place where it is oxidized by oxygen. So eventually everything will be then converted to NADH, and as we will see later, there is another one called coenzyme Q or CoQ that is also soluble in the membrane, and that's also oxidized by oxygen. So eventually whatever your oxidizing agents are, your reducing agent at the end must be NADH or QH2, which is CoQ. So all these vitamins together help to carry out this reaction, and this is the acetyl group, and in the process you are removing a carbon dioxide. So this reaction then is a decarboxylation reaction, and it is also a dehydrogenation reaction. We will see many times there the same enzyme it does both, or same enzyme complex does both. This is a complex of three enzymes. It does both. It's doing decarboxylation and oxidation. So in all those cases, the enzyme will be named as the dehydrogenase. So there are other examples also where it is a dehydrogenase, but at the same time decarboxylation also takes place. So this is one such example. The first one, thiamine pyrophosphate, again I'm not drawing the whole structure, just the important and relevant part of it. And there is more here. So this is the part that is important. This nitrogen, as you see, has 4 bonds. So that means it must have a positive charge, and that is very important that the nitrogen has a positive charge. So nitrogen is the electronegative atom. On top of that, it has a positive charge. So that means it is very electronegative, even more electronegative than oxygen. So it pulls electron from this carbon, and that makes this proton acidic. And if this proton is acidic, then in the mechanism, this will form an anion. Sulfur, nitrogen plus charge. So you form an anion here, and that is a very strong nucleophile, and that is what initiates the reaction. So as I said, we are not going to do the whole reaction, but the important part, so this is what initiates the reaction, this is what will react at this carbonyl group. So if this reacts at the carbonyl group, this is pyruvate. So this one reacts here and gives you this. And what it has done in this first step is to convert this to something that is similar to a beta keto acid. Remember why is beta keto acid decarboxylated easily? Because these electrons can go here. This one is also decarboxylated because you move this bond here, you move this bond here, and the electrons go to the positively charged nitrogen. So that's how the coenzyme works. Vitamin B1, it converts the alpha keto acid to something that is similar to a beta keto acid. Or in other words, something that is an electron sink helps to transfer the electrons from the carboxyl group to the nitrogen. Alright, so this will then decarboxylate, and then there are several steps. This is now converted to a 2-carbon compound. The 2-carbon compound needs to be oxidized. Remember, this is the oxidation step. So and so this is a decarboxylation step, and the enzyme is also called a decarboxylase, or as I said, the whole enzyme is together called a dehydrogenase complex. So the dehydrogenase complex has a dehydrogenase enzyme in it, yes.
What happened to the top double bond?
There's a double bond there, yes. Yeah, I'm not drawing the whole structure. There are more carbons in it, which I haven't drawn. But this is the important part in it. All right, so after this, then this will be oxidized. So you need an oxidizing agent. And that is done by the second enzyme. So this is a complex of 3 enzymes. And each of these enzymes are present in multiple copies. So basically there are about 24 different subunits in this whole enzyme. So together the whole enzyme is really a huge enzyme. And as I said, alpha keto acids are difficult to decarboxylate, and that's why it has all these structures. And not only this is just an example I'm showing. Later on in citric acid cycle there will be another enzyme that is decarboxylating a alpha keto acid, and that time I won't go into the details, but it's the same thing. Whatever this uses, that also uses all the same steps. Same thiamine, same lipoamide, same NADH if it uses.
The names of the enzymes?
The enzymes have names, we are not going to that, but the whole complex together is called pyruvate dehydrogenase complex. And if you want to know the names, we can go over that as we do the steps. So in this step, then the lipoamide is the oxidizing agent. It gets reduced. So not again writing the structure, so it gets reduced. So that means this hydrogen needs to be converted, needs to be removed to get it into the keto form. Remember, you are forming acetyl-CoA, so it must be a C double bond O here, so CH3CO. So this must be a ketone, so you need to oxidize that alcohol to a ketone. And that is done by lipoamide, which goes from oxidized to reduced form, and lipoamide never leaves the enzyme, so obviously it must transfer the hydrogen to something else. So basically you are transferring a reducing power to another oxidizing agent, which is a common strategy in the cell. Whenever something is permanently bound to the enzyme, it must transfer the hydrogen to something else. So that is the second enzyme, E2 let's say, and then it is transferred to coenzyme A. Again, coenzyme A has a very long structure. It has a nucleotide structure like adenosine phosphates, and then another vitamin, and then finally at the end there is acid group. So that's what is important. So it's coenzyme A is written as CoA-SH, and this is the important part. The sulfur is the one that reacts. Yes?
Is coenzyme A acetyl-CoA?
No, coenzyme A is coenzyme A. When it combines with acetyl group, that's called acetyl-CoA. So it will combine through the sulfur, and we did that in the previous chapter. It forms a thioester. Just like this is, if this was an alcohol, it will react with the acetyl group to give you acetate ion, oxoester. Instead of alcohol, this is a thiol. It reacts with acetic acid to give you thioester. So this will be S, then followed by OA, which is a very large molecule. So this is your thioester. So this will be the acetyl-CoA. And this is your final product. So this one, the acetyl group is transferred to Remember, this is the acetyl group, 1,2 carbons, and that is now oxidized by this, so that gives you acetyl-CoA. Alright, so these 2 carbons coming from here transferred to coenzyme A. And this transfer is easy to do because lipoamide, I haven't drawn the structure, I'll talk about that in a minute, that has SH in it also. So from one SH, it is transferred to another SH. So that's how you form coenzyme A, and this is what goes to the citric acid cycle.
Those two say the same thing, no?
Oh, what did I coenzyme A? Coenzyme A, and this is acetyl-CoA. This one. So acetyl-CoA goes to the citric acid cycle. But in that process, this lipoamide is reduced, and this reduced lipoamide needs to be oxidized again. As I said, anything that is a phosphatic group needs to be oxidized again. So that is done by the third enzyme, which is E3, which converts the reduced lipoamide to oxidized lipoamide. And so you need another oxidizing agent. And what is that oxidizing agent? And that is this part of this enzyme is another coenzyme is FAD. And so in the process, FAD is converted to, I should write it here. FAD is converted to FADH. Alright, so look at it this way, as it is oxidized, it's basically you're transferring the hydrogen. The hydrogens came from the pyruvate. It went to the lipoamide. Then the hydrogen from the lipoamide is transferred to FAD. That is part of this enzyme, coenzyme. FAD is also a prosthetic group, so it cannot just stay like FADH. It cannot be oxidized by oxygen, and so this will then be oxidized. I need more space here.
Wait, what does acetylcoenzyme A turn into?
Acetyl-CoA is not turning anything, it's going to the citric acid cycle. That is the end product. That's the goal of this whole reaction. Pyruvate converts to acetyl-CoA, and that goes to the citric acid cycle. So before it does that, we need to talk about the reducing agents, because that is what is going to give you energy. Always remember, your energy comes from the reducing agents, because they will be oxidized later to give you the energy. So this is one step where you are forming a reducing agent, and you need to make it into a useful form that can be oxidized by oxygen. Lipoamide cannot be, because it is in an enzyme, it never gives enzyme. That is transferred to FAD, that also cannot be oxidized, it is part of an enzyme. And so this has to be again transferred, converted to NAD. The FADH2 will be converted back to FAD, and NAD in the process makes NADH. So in all these steps, whether it is glycolysis, citric acid cycle, or this reaction, your goal is to make NADH, because at the end, the NADH will be oxidized to give you ATP. And this is how it is done, and it is done through so many steps, because this is a alpha-keto acid, and all alpha-keto acids are oxidized this way. Alright, so you got your NADH that will be later oxidized to give energy, and the one that I erased, let's write that again, that coenzyme A will give you acetyl-CoA. So these are the two products that we are interested in, acetyl-CoA and NADH, that will be used in the next step. Before we go on to the citric acid cycle, two things to remember. Acetyl-CoA will be going to the citric acid cycle, but two things to remember already. The pyruvate is first transferred from cytosol, from the cytosol to the mitochondria. This is where this reaction is happening. So I mentioned that last class. it has to be transferred from the cytosol to the mitochondria because this is an aerobic reaction. And that transfer is again another process which we are not going to talk about now. We will talk about that in the chapter on lipids because you need always some mechanism to transport things through the membrane. They just don't go in and out at random. So pyruvate has to be transported into the mitochondria. Secondly, this lipoamide has an important characteristic because this, as I said, has 24 subunit complex, and it is transferring basically the lipoamide is transferring the reducing power from here to the FAD. So you have a huge complex, and somewhere here is the lipoamide, and that is transferring let's say the hydrogen in the form of reduced form, and then it is transferring to the enzyme 3. So here is enzyme 2, here is enzyme 3, and this mechanism of transferring is called a swinging arm mechanism. The swinging arm basically refers to the lipoamide, which is actually 8 carbon compound. There are 8 carbons in a chain, and at the end you have the SH group. So let's say 1, 2, 3, 4, 5, 6, 7, 8, and you have the SH here and the SH here. And this part is attached to the enzyme. So as I said, this is a prosthetic group. It is bonded to the enzyme, doesn't leave. And then this is attached to the enzyme. When I say attached to an enzyme, obviously some amino acid in the enzyme. And that amino acid is actually lysine. So it is the lysine of E2. And lysine, as you know, has a 4 carbon side chain. So starting from the center of this to this part where the acetyl group will be, where is about 12 carbon long. And that's how it helps to move things around from one side to the other. So acetyl group is moved, when it is formed by E2, is moved from here to the coenzyme A that is catalyzed by another enzyme. So this type of mechanism you will find in all complexes, and the whole idea behind having large complexes is that you don't want the intermediate products to leave. And that process, when you don't want your intermediate products to leave, is called whenever you have a very large complex, obviously it is doing a complex reaction with multiple steps, so there will be intermediate products. And if the intermediate leaves the enzyme, then the enzyme will never find it again. It goes into the cytosol and becomes diluted. So all enzyme complexes work this way, that it doesn't let the intermediate leave, so it directly goes from one subunit to another subunit, or one enzyme to another enzyme. Thereby it increases the concentration of that. So this is the lipoamide that is helping in the metabolite channeling. If the acetyl group left, the enzyme is never going to find it again from the cytosol. So once it is formed in this enzyme, it is transferred from here to the third enzyme through this big arm that it has. Yes?
So what exactly does the lipoamide transfer?
The acetyl group. All right, so as I said, those are the 2 points to mention regarding this. And at the end, as I said, your end product of this reaction is the acetyl group, acetyl-CoA, and NADH. NADH will be later oxidized. We are not going to talk about that NADH, but that is the important goal of all these reactions. Yes?
What is enzyme 2 and 3?
Oh, they have names. They have very weird names.
Well, so does lipoamide is the coenzyme that transfers it to the enzyme?
Yeah, and it also gets oxidized and reduced. It has the SH group there. Yes.
So, one of the swinging arm mechanisms is to transfer hydrogen.
The hydrogen, as it becomes reduced, it is also It is also bonded to a acetyl group that is also transferred from one to another.
So it transfers to the acetyl group in its entirety.
Can you explain the swinging arm mechanism just one more time?
The swinging arm means, more important than the swinging arm is the metabolite channeling. That means you don't want the intermediates to leave, and the swinging arm is basically doing that job in order for the intermediate to go from one part of the complex to another part, which is a very huge complex. That also may not be a very feasible step to do. So that's why the swinging arm is taking it from one end of the molecule and to the other end of the molecule. And that happens because this has a 12 carbon chain. It has 8 carbons in the lipoamide structure plus 4 carbons of the lysine that it is attached to. That makes it a long chain, and so it is able to move within the complex from one end to the other end. And similar to that, other not so large complexes, but anytime you have a complex of enzymes, the idea behind having a complex is that you don't want the intermediates to leave, so that the reaction can happen more efficiently. And there will be many other examples of complexes that work together like that.
I'm just feeling.
All right, so this is the final reaction of this step in the process. You have removed a carbon dioxide. Remember, that is one of the goals of respiration, is to convert all the carbons of glucose to carbon dioxide. This is the first step where you have removed the first carbon. The glycolysis doesn't remove any carbon, it just breaks the glucose into 2 parts, 2-3 carbon compound. This is the first step where you are losing the first carbon. And at the same time, you are forming a high energy compound. So, high energy compound, this energy will be used to give you an ATP later. And at the same time, you are also forming NADH at the end. So, without going through the intermediates, lipoamide and FAD, the end product is NADH. And that is what is important. NADH will give you energy later, as I said, 2.5 ATP per NADH. And the high energy compound, that energy can be used for either or 2 purposes. One is to make ATP. Sometimes high energy compounds are used to make ATP. And as we have seen in the previous gluconeogenesis, for example, there is another use of high energy compound, and that is to just release the energy to drive the reaction forward. So those are the 2 steps, 2 things that high energy compounds do. Either they make a reaction possible by releasing that energy as making the delta G negative, or if there is enough energy in that, it will be used to make a reaction and make an ATP from ATP. And when that happens, that is called the substrate level phosphorylation. So this one then combines with oxaloacetate to give you the first product of the citric acid cycle. So oxaloacetate, oxalic acid is this product, this compound. This is oxalic acid, and when you combine that with acetic acid, you get this. So when you combine it, it won't be an acid anymore, so it will be a keto group. And so this will be your oxaloacetate. So the top part is the oxalic acid, bottom part is the acetic acid, and oxaloacetate is the 4 carbon compound, and this will combine with the acetyl CoA, which is a C2 compound, other than not counting the coenzyme A, which has many carbons in it. So this combines with this to give you, as an intermediate, it will give you citral-CoA. And then give you the citric acid. So that's the name of the cycle. So citric acid, so this will basically this carbons here combining with this one. So that will give you 3 carboxylic acid groups and citric acid. And I sent you the reactions by email. So citric acid cycle is there in any textbook, and it looks like a cycle just like this one. It's a very common figure shown in all books. And that with the first product is the citric or citric acid. So this is the citric acid. And once again, as I mentioned many times, all the carboxylic acids are present as carboxylate ions. So this is called citrate rather than citric acid. Yes.
I'm just confused, like where this falls in the citric acid.
The citric-CoA is combining with oxaloacetate.
So what's like that molecule that you have at the very beginning? Like, are you starting from the very beginning?
No, this is just from the previous one. The citric acid cycle. This is the acetyl-CoA. This is what happened in the previous step. So this is acetyl-CoA.
So what's before it?
And this is where we are starting the citric acid cycle. What is before it? This is pyruvate coming from glycolysis. Glycolysis gives you pyruvate. Pyruvate gives you acetyl-CoA that goes to the citric acid cycle. Yes?
And then you have oxaloacetate reactivation.
Yes, oxaloacetate is the first compound in the citric acid cycle, and as the name suggests, this is a cycle, and that means what you start with, you end with the same thing. Right, so this is a cyclic pathway, and you are starting with oxaloacetate, and you will end with oxaloacetate after all these steps. Alright, so 2 things to remember here. This is the first irreversible step in the pathway, and this is also the first committed step. Whenever you do a pathway, you must know which is the committed step, which is the irreversible step. This is the first irreversible.
Formation of citrate?
There is only one reaction here. What do you mean which step? There is only one reaction.
There's a lot of arrows.
Lot of arrows means this and this are combining. This one and this one are combining to give you citrate.
So formation of citrate is the first committed step?
Yes, right. And in that, so those arrows mean 2 reactants. 2 reactants are combining to give you citrate, and just to understand, oxaloacetate is C4 compound, acetyl group is 2 compounds, so citric acid is C6. So we are combining 4 plus 2 gives you 6. Now the citric acid cycle, many of you may have studied in biology. It is also called the cycle, right? And that's because Krebs discovered it, right? And when Krebs discovered it, no one believed him. He submitted the paper for publication, and it was rejected by Nature. Rejected by Nature, and that was his signature. He had that rejection letter posted on his door as long as he lived. Rejected by Nature. And only a couple of years back, Nature apologized to Krebs that they had rejected the paper.
I showed him that.
So that was discovered by Krebs, and so it's called the Krebs cycle. It goes by another name. It's called the tricarboxylic acid cycle, TCA cycle. why does the cycle have so many names?
Yes, I was. I've been talking about a lot of the things.
So from the Krebs cycle, it was called the citric acid cycle. And then people had confusion about whether there is really citric acid in that. So they were not sure. They knew there's something with 3 carboxylic acid groups there, so they were not sure about citric acid, so changed the name from citric acid to tricarboxylic acid cycle. And then they knew it is citric acid, and so the name is back again to citric acid cycle. Right, so what was the confusion? Why did people think that citrate cannot be one of the intermediates of this cycle? Because citrate is achiral. Right? Everyone sees that citric acid is not chiral, whereas all the molecules in the cell are chiral. And at that time, people did not know that you can make a chiral molecule from an achiral molecule in the cell. You cannot do it in a test tube. You learned in organic chemistry that whenever you create a chiral center, you are always going to get R and S in equal amount. Right? You cannot create optical activity, so how does the cell do it? You start with something that is not chiral and you are making chiral because citrate is not chiral and the rest of the products after that are chiral. How is that possible? People did not know much about chirality at that time, how the cell makes chiral molecules. And so there was doubt, and that's why the name changed, and then they changed it back because now we know how enzymes make only one chiral product. All the reactions that we have studied and any reaction you are going to study, most biological things have chiral centers in them. And the cell, when it makes them, it will not make a mixture, racemic mixture of R and S. It will make only one of them in 100% yield. And that is because of the enzymes. The enzymes themselves are chiral, so they hold on to the molecule in one direction only. And so that gives you either the R or the S. It will never give you a mixture of R and S. Whereas if you do that reaction in a test tube, you are always going to get a mixture of R and S. So that thing is settled, and that's why the name is back to citric acid cycle. What happens to this energy compound? As I said, the acetyl group, the acetyl-CoA is a high energy compound, so you don't want to just waste that high energy by hydrolyzing it. And that is exactly what it is doing. I didn't write the structure of this, but this has the coenzyme A here, and then that coenzyme A is lost. It's lost as coenzyme A. That means this bond is hydrolyzed. So there are certain situations in which high energy compounds are simply hydrolyzed without making ATP. As we will see later in citric acid cycle, there is another step in which there is acetyl-CoA. Not acetyl-CoA, the thioester, which is the succinyl-CoA, and that gives you ATP. So succinyl-CoA gives you ATP, acetyl-CoA doesn't give you ATP. What is happening here? And whenever this happens, always the reason is that this is being used to drive the reaction forward. One problem with all these cyclic pathways is that reaction has to be irreversible. That means the product has to be less than the reactant. So in every step, if the product is less than the reactant, when you complete the cycle, you are left with very little of the last product. So how is that going to start another cycle? So the amount of oxaloacetate is very low concentration. The cell maintains this at a very low concentration. If this is a very low concentration, then this reaction cannot be irreversible. Remember, for an irreversible reaction, you must have high amount of reactant and low amount of product. That's what makes it irreversible. But you are starting with a very low concentration of oxaloacetate because it's a cyclic pathway. You cannot always have high amount of reactant all the time. At some point there will be very low amount. And here there is a very low amount, but still this is an irreversible reaction. So how do you make that irreversible? Just by hydrolyzing that high energy bond, and that's what that energy released is what driving the reaction forward. So that's how the first step is irreversible, because that coenzyme A bond is hydrolyzed even though the reactant concentration is extremely low in this pathway. Alright, so we will stop here and then we will go over the rest of the steps. Yes.